So the next presentation, the topic is viruses are a major cause of amyloid. Epidemiological evidence indicates that antiviral treatments for severe herpes virus type 1 infections may confer protection against Alzheimer's disease and or dementia, and that may suggest that there is a causal role for the virus. And that's the topic of the current debate. And the yes position will be defended by Ruth Itzhaki, who is a physicist, a neuroscientist, an emeritus professor at Manchester University, visiting professor at the University of Oxford. And the no position will be defended by Magda Zolaki, who is a professor of neurology and used to be head of the first university department of neurology in Thessaloniki. We have to start with a vote again. So can I please ask the audience, do you think that viruses are important for amyloid deposition, or do you disagree?
Hello, everybody. I want to thank the organisers, first of all, for inviting me and say that I'm sorry that we're only virtual, all of us, but I hope the next time we'll all be there in person. Anyway, I'll start by talking about viruses, just in case there's some in the audience who are not very familiar with them. And I'll just say a little bit about their characteristics. Well, their main aim, of course, is replication, and they can do so only inside cells using the machinery of the cell that usually kills cells. Herpes viruses, though, have the special characteristic becoming latent, but they can be reactivated by stress, immunosuppression, inflammation, and so on. And reactivation causes productive infection, viral replication, as well as direct viral damage in the cells and inflammation, importantly. Infection effects vary, though, from asymptomatic to fatal. So you can be infected, but not necessarily affected. You can have some viral or bacterial illness, but it doesn't mean to say you're going to show any signs of it. You could be asymptomatic, in which case you might well be classed as a control. That's wrong, but that happens. And that's because you haven't shown symptoms, but there's another way of telling, if there's another way of telling you you're infected, that's the best thing to do, to make sure that you know who is and who is not infected. Anyway, herpes simplex HSV1 is the main agent implicated in AD, but other pathogens are probably involved too, but I don't have time to talk about those today. So we'll go straight on to talk about viral presence in brain. This was first detected, the viral DNA, in extracts of brain by PCR in 6 separate studies, firstly by my lab in 1991, after great difficulty in getting it published because people didn't believe it. However, this was the first microbe out of many to be shown definitively to be present in elderly human brains, and the presence we found depended on the brain region, age of subject. The prevalence was very approximately 60 to 70% in both elderly and AD brains. This might be an underestimate, we don't know. We were very disappointed, though, that we thought very naively there might be more in the Alzheimer brain, much more frequent, much more higher prevalence in the AD brains than in the normal controls, but that wasn't the case. So we decided there must be a difference in genetic factor between the two groups, normal, elderly, normal, and Alzheimer's. And so what we decided to look for was the ApoE genotype, because that was very much in the news in the mid-1990s. ApoE4 having been found to be a strong susceptibility factor for AD. So what we did was we looked by PCR at the allele frequencies, and what we found was a very high ApoE4 allele frequency in our HSV positive brains of Alzheimer's disease, but low frequency in HSV negative brains and in elderly normal brains. And what we could show from our results was that people were not getting infected with HSV1 because they had an E4 allele. That wasn't the case, nor was it the case that those who had AD were more susceptible to HSV1 infection. But what we did show was that the combination of HSV1 in brain in ApoE4 carriers conferred a strong risk of the disease. Well, we then had a rather speculative idea, which marvelous to say did work out. We thought it might be interesting to look at the allele frequencies of people who suffer from cold sores, herpes labialis, because these cold sores are caused by usually HSV1. So we did that, and we were very, very pleasantly surprised, I must say, to find that the ApoE allele frequency was much higher in the cold sore sufferers than it was in the non-sufferers. These were people who were infected. Everybody in this group was infected, bar one person. But these were people who were infected but didn't have any obvious symptoms. So as I say, it happens, and this has to be taken into account, which it often is not in epidemiological work. Well, we went on then to think about what are the links between HSV1 and AD. And the one obvious thing to look at was whether or not HSV1 infection of cells in culture produces characteristic features of Alzheimer's brain. And so what we looked for, we looked in cell culture for beta, whether or not A beta was produced, amyloid, and whether or not abnormally phosphorylated tau was produced. And these show the results very briefly. These are infected glioblastoma cells. This is infected, stained for A beta, and this is the uninfected, but as you can see, shows absolutely nothing compared to that, or very, very little. This is HSV-infected mice, brain from those animals, and this is stained for A beta, and these, in fact, are about plaque size, or which is of course variable, but more or less on average about plaque size. This is uninfected mice, brain from uninfected mice, which, as you can see, shows nothing. And here we have staining for p-tau, phospho-tau. We looked at about 4 regions by different antibodies, and this is the sort of staining we found. This is uninfected human glioblastoma cells, and as you see, very little there. So this was a nice link. By the way, I keep referring to our work simply because we were about the only people doing this work for the first 15 years or so, starting from about 1990. Other people then started joining us, particularly in the last 10 years. There's been a big surge. Anyway, we went on to a much more speculative idea to find using In-Situ Pcr, which actually locates target within tissue. We look for HSV1 DNA in brain tissue and saw how it related, if at all, to the localization of plaques. And to our, again, surprise and delight, we were very lucky, but not always so, of course, but in this case we were, in that this is what we found. This is HSV1 DNA stained by In-Situ Pcr. These are plaques stained by thioflavin S. And this is the merge. And as you can see, there's really complete localization coincidence of the two. This is another tissue slice specimen, and this shows staining of A beta by immunohistochemistry. So it's the same sort of idea. The two, brown and purple, overlap. And most of the DNA in the case of AD plaques, or AD brain plaques, is present in plaques, but only 24% in age normals. So this was a very nice finding, and we thought this might mean that the amyloid acts as an antimicrobial agent that sort of tries to encage the viral DNA. We then did one other series of experiments with various antivirals, in this case, acyclovir, which is a common one. And what we did was we infected cells and then treated them with increasing concentrations of acyclovir and stained them after that for these various aspects. This is staining for HSV1 proteins. As you see, it goes down, not to zero, but there's a good explanation for that. This is staining for A beta, and again, as you see, it goes down considerably, but it doesn't flatten off, probably continuing at higher concentrations. However, in the case of phospho-tau, it went down to zero, and this is very interesting and useful, and it made us fairly feel quite convinced that acyclovir or other antivirals should be used to treat Alzheimer's disease patients. So we can, from all these results, we listed the HSV1 AD concept. This first one you've seen, there's HSV1 in latent in brain, and many elderly people can reactivate HSV1 in brain, and genetic factor E4 confers strong risk of AD, and ApoE4 is a risk for herpes labialis. And now there are over 400 studies using very diverse approaches which support HSV1's role in AD. So what's the general evidence amongst these 400 studies? Well, firstly, very interesting result relevant to my present work: systemic infection causes cognitive decline and leads to increased risk of AD. Cognitive decline is greater in E4 HSV1 infected humans than in E2 or E3 carriers, and genetic support for a viral role, the link pathways found. IgM presence in serum indicates recent HSV reactivation, shows increases in dementia risk. And other evidence, we've already talked about A beta accumulation in cells and location of HSV1 in plaques, and tau accumulation caused by HSV1, and also APP, amyloid precursor protein, is associated with virions, transport along axons. Synaptic function is reduced on infection by A beta accumulation, and A beta has an anti-HSV1 activity consistent with HSV1 location entrapped within plaques. So I'd just like to show you a few other things. Population epidemiology studies from Taiwan show that HSV1 infected people had a greater risk than uninfected of developing dementia, which was very interesting. Even more importantly, treatment of HSV1 infected people with antiviral drugs, that's well before any had developed dementia, if they were going to develop it. This greatly reduced the risk of dementia. And there have been 7 other studies since then, at least 7, which found a similar protective effect. And this strongly supports a causal role for HSV1, not just an association. Then there was another very important paper which showed that in 3D brain models, doing simply HSV1 infection causes A beta formation, also p-tau, which isn't shown here, and gliosis, inflammation, synaptic damage, all phenotypes of AD. So this was very strong support again for HSV1 having a major role. And just to mention briefly what we found recently, we looked at several vaccines have been found to reduce AD dementia risk, even when they had nothing to do with AD or dementia. Vaccines were completely apparently unrelated diseases. We found that in the case of shingles, it causes a slight increase in risk of dementia, but Zostavax vaccination against shingles reduced the risk greatly of dementia. So we had already asked in 2001, as a result, 2002, a result of a paper by Vero et al., which we suggested that infections increase dementia risk by reactivating latent HSV1 in brain via neuroinflammation. And we also suggested that vaccines might reduce the risk by reducing the severity or the number of infections. So this turned out to be what we've shown now is completely consistent. Next slide. This HSV1 infection, as we have said, causes AD phenotype. VZV infection we've examined, and that does not cause accumulation of beta and p-tau, but does produce gliosis and inflammation. VZV added to quiescently infected cells latently affected in effect with HSV1, causes the AD phenotype to appear. In other words, it reactivates the latent HSV1, which we thought was a very, very interesting result. And this is totally consistent with infection causing HSV1 reactivation, which we suggested, as I said, in 2002, and consistent also with a very interesting review by these people who cited a number of papers which showed that BCG vaccination reduced cold sore frequency and severity, even when they had nothing to do with HSV1, for which there, unfortunately, is at present no vaccine. Obviously, one hopes in the future that there will be, and one hopes in the future that antiviral treatment will work to prevent, hopefully, Alzheimer's. That would be wonderful. I'll leave it at that, and thank you very much for listening. Just show the next slide. Very small number of people who've worked in my lab, but a large number of collaborators who've helped enormously.
Hello to everybody. I want to say thank you to Professor Amos Kortsin for this kind invitation. And these are my disclosures, and this is the outline of my presentations. And let's say some words about introduction. Certainly, the amyloid cascade hypothesis partly explains the immediate cause of Alzheimer's disease. However, studies on familial and sporadic Alzheimer's disease have shown that amyloid beta is the initiator of AD rather than the accumulation of tau protein. My search in PubMed some days before showed that there are more than 54,000 papers about pathogenesis and Alzheimer's disease. 10% of them are about inflammation and Alzheimer's disease, 10% of them are about bacteria and amyloid deposition, and only 370 about viruses and amyloid deposition between 1969 to today. This is a very simple figure about the pathogenesis of Alzheimer's disease. You can see that we have transmitter dysfunction, cholinergic, monoaminergic, glutaminergic. We have metal ion dyshomeostasis. We have vascular disease involvement, oxidative stress, mitochondrial dysfunction, and finally we have also inflammation. And these have also, as a result, neurofibrillary tangles and amyloid plaques, and the end of all these pathogenetic mechanisms comes to neuronal death. If we take only amyloid beta peptide, we can see that it leads to reactive oxygen species, and this lead to metal ion dyshomeostasis, mitochondrial dysfunction, and neuronal apoptosis. Also, it is responsible for altered neurotransmission, as we discussed before, and synaptic failure. And we have also neuroinflammation via reactive microgliosis and astrocytosis. And also we have the pathway for amyloid plaques and neurofibrillary tangles. If we see recent studies about the pathogenesis of Alzheimer's disease, we can see that the damage of DNA is the recent program which explains the pathogenesis of Alzheimer's disease. And we can add to the other pathogenetic mechanisms the genomic instability, telomere attrition, epigenetic alteration, loss of proteostasis, altered intracellular communication, stem cell exhaustion, cellular senescence, mitochondrial dysfunction, as it was also before, and deregulated nutrient sensing. And these are the 9 hallmarks of aging or diseased brain. And if we would like to see what happens with the cells, the different cells in our brain when we have Alzheimer's disease, we can see how neurons become, how astrocytes, microglia, oligodendrocytes, pericytes, vascular and endothelial cells, all the cells are destroyed during Alzheimer's disease neuroinflammation. And if we accept that Alzheimer's disease is accelerated aging, we can see that aging has also 6 different pathways. Between them is inflammation, and because of inflammation, we can say that viruses and bacteria are involved in pathogenesis of Alzheimer's disease. And also we have a combination of mitochondrial dysfunction and neurogenesis in neurodegenerative diseases, and this combination and many other combinations can lead to Alzheimer's disease also. If we would like to have a figure for all these mechanisms, we can discuss about genetic one and environmental one. And the mutations are responsible for familial Alzheimer's disease, 1 to 5% of cases with early onset of disease. And also risk genes, there are about 38 risk genes until now. And environmental risk factors are hypertension, smoking, diabetes mellitus, diet, education, and many others, as we can see in other presentations. So we can see we can say some words more about the genes that are related with Alzheimer's disease. And as we can see in this table, we can see causal genes, which all are related with amyloid. And risk genes, from these 11, the most frequent, 2 are involved with innate immunity expressed by microglia and immunoresponse. And also a recent study 4 years before involved also not only 2 of the 11 I discussed as before, but also other risk genes which are involved with microglia function. So they are also responsible for the response to viruses or bacteria. And also we have evidence about the results of anti-inflammatory drugs, and also we have many anti-inflammatory agents which are in clinical trials now for Alzheimer's disease. We have also other genes which are responsible for Alzheimer's disease. This is a recent study of our group from EMIF project. And we have also some information about C-reactive protein levels in more than 100,000 individuals from general population, which support the idea that Alzheimer's disease is not an inflammatory disease, because low plasma levels of CRP at baseline were associated with high risk of Alzheimer's disease. And also we can add in genetics mechanism the epigenetics inheritance processes, because we can see that there is a DNA methylation, and as you can see, the levels of these risk genes or proteins are increased or decreased. Also, there is another epigenetic mechanism, histone modification and microRNA changes. All are changed during Alzheimer's disease pathogenesis. And there are not only these epigenetic mechanisms, there are also other epigenetic mechanisms which are described in recent papers. You can see here from November 2021. And let's go very, very quickly to environmental risk factors, which are also not correlated with viruses and bacteria. There are 2 studies which were published the previous year, which showed that the first one, a meta-analysis of 296 studies, which showed 10 certain risk factors, as we can see here, and 9 class 1 factors demonstrated level B evidence. That means that we have to do more studies in order to discuss about this. And there is also a study by the Lancet Commission, which added in the previous certain risk factors other 4 risk factors, as you can see here. And also they discuss about protective factors and also other new risk factors. Between them, there are infections, and also perhaps in future we'll see that COVID-19 is a risk factor for Alzheimer's disease. And let's see some environmental risk factors which are not discussed in many papers, in order to see how they lead to Alzheimer's disease, and they are not connected with bacteria and viruses. Oxidative stress can lead to age-related hearing loss, and this can lead to frailty syndrome, which also comes to Alzheimer's disease, but also only age-related hearing loss can lead to Alzheimer's disease. And we can see this picture also, which from age-related hearing loss, we have frailty syndrome, which is a combination of physical inactivity, emotional problems, and cognitive problems, which lead also to Alzheimer's disease. Also, there is much discussion about healthy diet. And recent studies support the idea that we have to do much work with ketogenic diets in future. And also we have information that these different diets have to do with methylation of DNA or other epigenetic mechanisms. We can see an example here. Diet has an effect on microbiota here, and this changes the DNA methyltransferase, which also lead to alter gene expression, and this comes to different mechanisms which lead to Alzheimer's disease. And we can see how different diets, as you can see here, do different changes in methylation patterns, as you can see here. And if we take only short-chain fatty acids, we can see that they have multiple roles, but also between them, between 5 different roles, they have also modulation of neuroinflammation. So we have to do much work in order to see if this kind of diet is involved in the risk factor from bacteria and viruses. And let's see a big catalog of neurotoxicants which are related with the pathogenesis of Alzheimer's disease. You can see here all these neurotoxic agents with different mechanisms lead to Alzheimer's disease. And we can see this very simple picture, which shows that if we stop this mechanism from these neurotoxicants, we have no amyloid plaques, no fibrillary tangles. We have increased synaptic plasticity, spatial memory formation, cognitive function, increased cognitive function, and so on. So my conclusions from this study, which was published in August 2021, we can see that now there are many clinical trials ongoing. Between them also there are clinical trials about inflammatory cytokines and neuroinflammation. And also there are many trials, clinical trials with anti-inflammation drugs. You can see some of them, and you can see here antibiotics and also antiviral agents, which can show some results. So we have to wait in order to support the idea that Alzheimer's disease, one of the main risks of Alzheimer's disease are viruses and bacteria. But we have not to forget that also tau protein is related with immunity, and we have to discuss and see the connection between tau protein and viruses also. Finally, I would like to show you that in this year we had for the first time that we have Alzheimer's-like signaling in brains of COVID-19 patients. And perhaps we don't know yet the mechanisms, but of course we'll learn more about these mechanisms in future. And my conclusion is first that there are many causes which are related with amyloid deposition and tau deposition. And second, bacteria or viruses are not the main cause of amyloid deposition, but we need more research in order to say that is one of the main causes. Thank you very much from our lovely CP Thessaloniki.
Move to discussion now. I hope the audience can hear us. So thank you very much for the presentations, both of you. Ruth, Magda, do you have any immediate reactions on the other presentation? Please, yes. Ruth.
Firstly, thank you very much for your nice talk, Magda. I think we don't actually disagree, because what our results suggest is that something like 50 to 60% of people, Alzheimer's patients, are affected because of viruses and maybe other infectious agents, but we know nothing about the other 40%. So the causes that she's suggesting could well be the case. They might, the other factors she's suggesting could well be the case, the cause of the disease. So we're not claiming for one moment that herpes or other viruses or bacteria are the cause. We think they are a cause and a major cause. What I do think also is that neuroinflammation is very, very important. It seems to be a factor in a lot of very many studies. And we think in our recent, I think, very exciting work, where we show that VZV reactivates quiescent or latent HSV1 in cell culture, in 3D cultures. We think that's probably mediated by neuroinflammation, which is VZV causes inflammation. And we think the cytokines that it causes are those that are actually reactivating the HSV1. Whether or not that happens with other infections, we don't know, but we're just about to set out to find whether or not they do. I think that's all. Thank you.
Thank you, Ruth. There are two questions from the audience, or from the same person, from Jonathan Kennedy. So he wants to know, can you share whether the distribution of AGES1 in brain mirrors the pathological spread of Alzheimer's pathology? So is there a spatial overlap of virus and amyloid in the brain of Alzheimer's disease patients?
Well, the closest we've done to that are the in-situ PCR studies we did, where we looked in tissue sections by in-situ Pcr, which is Pcr done on sections as opposed to extracted DNA. And what we showed was there was very, very good co-localization of HSV1 DNA, literally within plaques. A very high proportion of the DNA molecules there were located within plaques. So this was true in AD patient's brain, but to a much, much lesser extent in elderly normals. So yes, we think there is a great deal of co-localization. And of course, in herpes simplex encephalitis, one of the main reasons that we looked for evidence of HSV1 acting in brain was because the regions affected most in herpes simplex encephalitis are exactly those affected most in Alzheimer's disease.
And there's another question for you from Jonathan Kennedy. So he wants to know, do you think that there are any parallels to be drawn between your work and the occasional development of anti-Nmdar encephalitis after HSV1 infection?
Oh, I don't know. Sorry, I don't know. I'd have to think about that. I don't know the answer.
Good. And Magda isn't here, is she? Can't see her. No, but Amos is. Please, Amos.
Yeah, thank you very much for the talks. Very nice. I would like to ask you, Ruth. So you showed animal experiments and the in vitro cell experiments, which showed that actually there is A beta production, but A beta production can be seen also as a biomarker, perhaps not specific acute stress response. So this does not really necessarily show that its relationship to Alzheimer's disease. Now, all these animal and viral studies are, of course, short-term. And even if we accept that the herpes infection of the human brain can cause all these things, then we need also to explain what happens during the latent period. So it takes about 50 years maybe from the time that we are first exposed to herpes and until something erupts as a neurodegeneration. So what do you think goes on there?
Well, I think one reason why that is the case is that probably the virus doesn't get to the brain from the periphery, the trigeminal ganglia, or maybe as a new infection through the nose. We think that probably happens in middle age. And so whatever's happening in the periphery might not be really relevant to what's happening in brain years later in older age. In other words, the virus starts it becomes latent in brain when it's traveled there middle age, and then events such as immunosuppression or infection or stress generally reactivate it, so that the damage occurs in older age, not in brain, but not in, I mean, we looked for virus in brain of younger people and we found it present in only a very small proportion. So I think that suggests that there must be some movement or some introduction of the virus to the brain in round about late middle age.
So what happens at middle age? Do you think that there is reinfection and therefore peripheral response, and how does it get to the brain? Do the viruses get to the brain or what?
Well, there's some evidence that it could be a new infection through the nose along the olfactory pathway, but there's also, I think, evidence that it comes from the periphery, traveling axonally. So I think it's uncertain what happens. It could be both or either. I don't know. Or it'd say, I don't know, it's not known.
So how are we going to proceed? Because it's important to know whether we want to prevent reinfection or to prevent the effects of reinfection or the effect on the brain, or these are critical steps that you theorize, but we need to know in order to move forward.
Well, there are 2 points. Firstly, antiviral treatment was shown in the Taiwan study and by 7 or so other studies recently. Treatment for herpes, serious herpes infections has been found quite extraordinarily to reduce the risk of Alzheimer's disease. Why or how that is, I don't know, because these antiviral agents are short-lived. But if that is the case, then that points the way to prevention. I was thinking what the other thing is.
And do we know if those antiviral agents have any other non-specific effects which are not targeted at the viruses? I mean, many other medicines have a direct effect on amyloid, even though they're antidepressants or antihypertensive. So do we know anything about kind of non-viral effects of antiviral agents?
No, that's a very important point. I mean, I agree with you and been thinking about this, where I've looked up the literature and can't find anything that's relevant, but it's something I very much hope to work on, because I think this could be the answer. Is really peculiar. There's also a recent study, interestingly, which shows that lithium seems to have an effect well before any Alzheimer's traits are shown. So that seems to have also a long-term effect where one wouldn't expect it. But I don't know. I haven't read the paper properly yet. It's only just seen. I've only just seen it. So we'll find out. The other thing is about ways of finding out. Well, one way, which is rather crude, but it's the only thing that could be feasible, is if one could get hold of CSF from people of a range different age groups. The point is, I didn't mention it in my talk because there wasn't time, but we found that the virus is not just present in brain dormantly, but it is actually doing something, because antibodies are present to it in the CSF, which suggests that it has been active there. It's well known that they stay quite a long time after herpes encephalitis, which is why we look for antibodies to HSV1 and CSF. Well, if one could look at CSF, and this is the problem, I haven't been able to get hold of CSF from people of a different ages. But if we've got CSF from people, say, from age 20 to 30, 30 to 40, 40 to 50, I'm not expecting them all to show this, but those who do show antibodies in the CSF, one would expect on this basis to find a far higher proportion of people who have these antibodies in the older age groups. So it's a question of finding people, and there are, I think, stores of such things, but I don't know how to find them easily, people who have CSF collections. And if one could separate them into different age groups, this might perhaps show tell us something about when it gets to the brain.
And there's not only an association between viruses in Alzheimer's and dementia risks, but also for certain bacteria. So is it,
yes.
I mean, again, the question about specific and unspecific effects, could it be that just kind of the viruses and bacteria, infectious agents activate the immune system in the brain? And that has an effect on dementia risk. And it's not the virus, it's kind of more general activation of the immune system.
No, I agree with you totally. It may be that more people have a virus, HSV1 in brain. That, I mean, it looks like at the moment people are discovering a positive menagerie of infectious agents in brain. So, but of course, whether they're active or doing anything is a very different matter from finding them there. I do think that some bacteria, perhaps particularly Borrelia and Chlamydia pneumoniae, and maybe some of the gum disease ones could well be having an effect. And in fact, I have two colleagues who are doing work on this in collaboration in the States, looking for in individual brains for the presence of HSV1 and or Borrelia and or chlamydia and so on, to find whether each acts individually or whether there are two agents present in any one brain, or even 3 or 4. In other words, they act individually or in combination or what. And I think this surprising that hasn't actually been done as far as I know, but it could be very informative from the point of view of treatment and possibly prevention. So thank you for bringing that up.
Thank you.
But this is a no-winner because we know that, for example, periodontal diseases are a risk factor for Alzheimer's disease as well. So if it's not specific to any given infection, then and we are all exposed to pathogens, to bacteria, to viruses, and whatever all the time. So what you're trying to tell us that this is specific to a given virus, to HSV1 or maybe ZVZ. But I think that one important point that needs to be mentioned is the fact that after herpes encephalitis, some people develop anti-Nmdar antibodies, which have, of course, all cognitive effects as well. In some patients, it's just cognitive decline that occurs there. And this is, unlike what was mentioned before, this is particularly true for children.
Well, yes, that's a very interesting point. But about your earlier point, if one, obviously, the fact that one's infected with something does not necessarily mean that one's going to be affected, as I said much earlier in my talk. If that were so, about 80% of us would be developing Alzheimer's disease, because at least 80% of people are infected with HSV1. So obviously other factors, such as genetic ones, are responsible possibly for determining who is severely affected and who is not. And in the case that we looked at, it seemed to be ApoE4 in combination with HSV1 in brain that is responsible for in producing a high proportion of cases of Alzheimer's disease. This is probably true of bacteria as well and other infectious agents. So it's not a question of having the infection in your body, it's a question of what it does to you, what harm it does, or how less well it's repaired, the damage, I mean.
Mm-hmm. Okay, great discussion. I think we need to move on and we'll have another vote on this discussion. I hope everybody can hear us again. And now it's 40% yes, so in favor of CAN or our viruses, the main cause of amyloid deposition, and 60% think no, they're not. Thank you very much. Exciting discussion, very good presentations, and let's move on to the next debate.